Dr. Oyam Revach from MGH Cancer Center from Harvard Medical School. Oyam, the stage is yours. I don't know if we can hear you right now.
Can you hear me now and see my slides?
Yeah, great. Yes.
Yeah. Thank you, Shimrit, for the introduction and for organizing this wonderful symposium. I want to thank the organizers for giving me the opportunity to present my work. Today, I'm going to share with you a story that is currently under revision about targeting CD38-driven T cell dysfunction to restore sensitivity to immunotherapy. The major motivation for this project is that cancer immunotherapy can really cure metastatic cancer disease. And this is based on blocking the PD-1, PD-L1 axis and activating lymphocytes to fight cancer cells. Overall, about 50% of all cancer patients are eligible to some sort of immunotherapy, but unfortunately, only a small fraction of them respond to these therapies and have durable responses. And there are many factors that are dictating this, but one major one is T cell dysfunction. So T cells are undergoing TCR stimulation in the tumor microenvironment, as well as exposed to immune suppressive factors, overall making them less and less effective over time. So the goal was to uncover mechanisms of T cell dysfunction in human cancer and leverage them to improve response to immune checkpoint blockade. In 2018, Moshe Sodeh Feldman and Karen Yitzhak published this paper in Cell, where they did single-cell RNA sequencing of melanoma tumors that were treated with immune checkpoint blockade. What they showed is that tumors that responded to immunotherapy were enriched with this subset of effector memory-like T cells that do not express exhaustion markers and express the transcription factor TCF7, whereas the ones that were resistant were enriched with this subset of CD8 T cells that were in exhausted state and expressed multiple exhaustion markers. The classical immunotherapy is targeting one of the axes on those T cells, which is PD-1, and we chose to focus on another axis on those T cells, CD38. CD38 is an ectoenzyme that catabolizes NAD, and it limits the NAD levels in the cells, which can affect greatly on many cellular processes. Looking at the CD8 T cell clusters from human melanoma, we see that CD38 is mostly expressed in these three clusters of T cell exhaustion, together with PD-1, and anti-correlates with the expression of TCF7, which is mostly expressed in these three clusters of effector memory and early activated T cells. Most importantly, CD38 expression in T cells highly correlate with lack of response to immune checkpoint blockade and can even predict it. We validated those findings in another cohort of melanoma that was unpublished and another cohort of non-small cell lung carcinoma that is another type of solid tumors that are treated with immune checkpoint blockade as a first-line treatment. Overall, showing that CD38 expression in CD8 T cells highly associated with resistance to immunotherapy. So what CD38 is doing in those T cells? To imitate what we see in the tumor microenvironment, we did in vitro chronic stimulation of T cells. Either we stimulate them once to form acute stimulation, or we are stimulating them every few days for about 2 weeks to form chronic stimulation. The chronically stimulated T cells have high CD38 expression, and they also express high other exhaustion markers like CD39, PD-1, and TIM3. They lose their proliferative capacity and their cytotoxic ability, overall recapitulating what we see in T cell exhaustion. When we knocked out CD38 using CRISPR and chronically stimulated the T cells, we were able to maintain the proliferative capacity as well as their cytotoxic ability and the expression of TCF7, overall suggesting that these CD38 inhibited T cells are more like effector memory T cells. So next we asked what are the downstream effects of CD38 function, and for that we did gene set enrichment analysis of both human and mouse melanoma CD38 high T cells. We found few gene sets that were enriched. One of them is oxidative phosphorylation that is indicating regulation of mitochondrial function. Indeed, when we looked at CD38 high versus low T cells, we saw that the CD38 high have higher mitochondrial mass and membrane potential, which were both shown to be associated with terminally exhausted T cells. When we knocked out CD38, we were able to reduce the mitochondrial mass and membrane potential, recapitulating more effector memory-like phenotype. Because CD38 is an ectoenzyme, we next asked what are the global effects of blocking CD38 in chronically stimulated T cells. For that, we collaborated with Dr. Nama Kanarix from Boston Children's Hospital and did metabolomics of chronically stimulated T cells that were treated with CD38 inhibitor. What we saw is that there is a restoration of the NAD pools in the cells, so both NAD and NADP are restored when CD38 is blocked, together with the mitochondrial phenotype, and specifically the mitochondrial reserved capacity, which is indicating that the cells can respond better to increased metabolic stress, which is very important for T cell function. These cells had lower exhaustion markers and higher TCF7 levels, overall suggesting that the restoration of the NAD pools is restoring a better effector T cell function. So we were able to get a better functional T cells. So the next question was, can we use it to overcome resistance to immune checkpoint blockade? And in order to answer this question in clinically relevant models, we used our PDOTS model, the patient-derived organotypic tumor spheroids, where we take tumors from patients and we are mechanically and enzymatically degrade them to form these mini spheroids that contain the tumor cells, but also all the tumor microenvironment, including the immune system. We culture those in microfluidic devices inside matrix, and we treat them with immunotherapies that are used in the clinic. In this case, we use pembrolizumab to block PD-1, daratumumab to block CD38, or the combination. Daratumumab is a monoclonal antibody that is used in the clinic for multiple myeloma treatment, and here we used it in the context of solid tumors of melanoma. So after a week of culture, we assessed the viability of these spheroids using microscopy viability assays. Using this method, we collected a cohort of 27 melanoma tumors, and these are waterfall plots. So anything above the zero is tumor growth, and anything below the zero is tumor shrinkage. And we can see that to the PD-1 blockade, we almost do not see any response. And this probably has to do with the fact that most of the cohort is composed of clinically resistant PD-1 patients that are refractory melanoma. In the daratumumab treated ones, we see some responders, but with a combination, we see over 50% of responders, which at least half of them are clinically resistant to PD-1, suggesting that CD38 blockade can both enhance the immunity of the tumors and also overcome resistance to immune checkpoint blockade. And this is how it looks like in the devices. This is an example of a Merkel cell carcinoma, another type of skin cancer that is treated with immunotherapy as a first-line treatment. And this is a clinically resistant one. So we see that to any of the single treatment, we don't see any tumor immunity, but only with the combination, we see tumor killing. And this is how it looks in the devices. We stain them with live dead dyes as well as nuclear dye. And only with the combination, we see this spheroid shrink. So daratumumab is clinically approved for other indication, and this is great, but it's also a monoclonal antibody and can work in internalization of the receptor mechanism and also in Fc receptor mediated mechanisms like ADCC. So we wanted to verify if blocking the enzymatic activity of CD38 and increasing the NAD levels is sufficient to restore the sensitivity of the tumors. So for that, we collected another cohort of melanoma PDOTs that were treated with CD38 inhibitor, and we see both single agent effect as well as combinatorial effect with PD-1 blockade using the inhibitor of CD38. And to circle back to the role of NAD in all of this, when we looked at semi-responding tumors like this one that responded somewhat to the combination but not significantly, we were able to increase the tumor immunity by adding NAD to the media, underscoring the role of NAD in this process. So to summarize, we have these dysfunctional CD38 high T cells in melanoma that are highly correlated with lack of response to immune checkpoint blockade. CD38 is elevated on the T cells by TCR activation, chronic TCR activation, and also type 1 interferon signaling from the tumor microenvironment, which I didn't have time to show today. But CD38 overall is reducing the NAD levels and the mitochondrial function of those T cells. And when we block both PD-1 and CD38, we are able to maintain this effector memory-like phenotype that have increased NAD levels, increased mitochondrial function, and gene transcription associated with response, and overall overcome the response, the resistance to immunotherapy. So with this, I would like to end, and these are all our collaborators and funding, and to the patients and their family, and thank you for listening. I'll be happy to answer questions.
Thank you very much, Oriam. Very interesting. Do we have any questions?
I cannot.
I have.
Oh,
Yeah.
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 Yeah, very nice. Good evening. So PD-1 obviously has a ligand. Does the CD38 have a ligand? I think that there is some stories about CD38 binding CD31.
Exactly, on endothelial cells, but it doesn't seem to be the case here. So it has an ectoenzyme effect without the ligand.
Okay.
So we don't think we need the ligand for this story to work.
I have a question. What about other cells in the microenvironment of when you block CD38? Do you see any effect on the other cells?
Yeah, so that's a great question. So CD38 is expressed on other immune cells, as we know, but none of them is correlating with the response to immunotherapy. And when we looked at in vivo data after blocking CD38 plus PD-1, most of the populations that were affected were T cells, either CD8 or CD4s were increased in their tumors. So,
But do you see any effect on the other immunosuppressive cells in the microenvironment? So the microenvironment remains immunosuppressive, or you see any
yeah, we
effect?
think that's a great question. We don't know what's happening in PDOTs because it's very hard to profile after the treatment. But in mouse tumors, we see some changes in myeloid population, which are not super significant with the treatment. Most of the
It's
effect is on
mainly
the T
the
cells.
T cell population.
Yeah.
Do you see any effect on interferon gamma expression or granzyme B expression or any direct effects of cytotoxicity?
Yeah, so actually, I didn't have time to show, but when we did, we were asking like, what are the tumoral features that will dictate this response in human? And we did a single cell analysis on the CD8 T cells from the same tumors that we profiled ex vivo. And what we see is that there is enrichment of both granzyme A and interferon gamma in the responders, which suggests actually that this is not reversing exhaustion, but rather keeping those T cells for being active along the treatment time. So we need those population of granzyme and interferon gamma to work.
Hi, Orion. Beautiful work. What about CD4 cells? What about also TOX, one of the main master regulator of exhaustion?
Yeah, so I don't remember if we were looking at tox specifically, but the data from melanoma show that the TCF7 high T cells have lower tox. So it would be nice to see if the transcription changing there. And about CD4s, actually, the effect is more or less the same because when we work on CAR T cells, we look at a mixed population of CD4 and CD8s. And so in the paper, we have both TILs that are isolated CD8 T cells, and these are the effect. But also when we have the mixture, the effect are more or less the same, more TCF7, more cytotoxicity, and better function overall. So also for cellular therapies, the blocking CD38 can be beneficial, I think, in the expansion protocols, of CAR T cells and TIL products, as well as after in the in vivo setting.
Thank you.
Any other questions? All right, so thank you very much, Oriam.